First, we need the tissue, means nerve and muscle, and that should be in a living condition. The animal may die, but the cells may have to remain for certain in a living condition for certain period of time. Okay, so how we can manage that one for a certain period of time, that we will see. Then we need what? We need a setup for stimulus. Then another setup is needed. The muscle will be contracting, that should be recorded. So another setup is required, that is recording setup. So with this setup, we can demonstrate this experiment. Muscle, if appropriate amount, appropriate quality of stimulus, electrical stimulus is given to the nerve, this will be conducted to the muscle and they will be responded by making a twitch, means by contraction, then it will be relaxed. So this twitch means contraction of the muscle and its relaxation can be recorded on a moving drum. If a writing facility is available, the movement, one thing will be with contraction, it will be lifted. And when contraction is done, it will be relaxing, then again it will be coming down. So by this time has elapsed, so it will make a graph. This is called simple muscle twitch, simple muscle contraction. Okay. So let us first see how we can make a nerve muscle preparation. So for this purpose we need, how we will prepare nerve muscle. The first thing that we have to do is preparation of nerve muscle. That is gastrocnemius, sciatic nerve preparation. So we have to take a live frog, then we have to stun its head on a hard surface or with a stunning rod. It will become senseless. Then what will be done? So you can see this is stunned. Now at the back of the head there is one area, I forgot this area, here it is. And from there one needle is inserted to destroy the spinal cord. It will be passed through the vertebral column. So it will, within the vertebral column there is spinal cord, so it will be destroyed. So no outgoing impulse will be going, no incoming impulse, so it will be paralyzed. And it will be, the needle will be inserted towards it, then needle will be moved like this so that brain is damaged. Okay, then you dissect the animal to get this. So this is the leg and this is the sciatic nerve. This sciatic nerve originated from vertebral column. Okay. So some portion of the vertical column has to be retained, isolated, and this link should not be destroyed. So this link is with the vertebral column is there, and this is the sciatic nerve, this is the knee, and this is the gastrocnemius muscle, and this is the tendon Achilles. So now this part is removed. So after that, what we will get? We will get this. So this is the tendon, this is the muscle, this is the nerve, sciatic nerve, and this is the vertebral column. From here it is, the sciatic nerve is originated. So now we got sciatic nerve, gastrocnemius muscle preparation. Now, if it is isolated, now it has to be kept in a condition where the cells may remain alive. So for that purpose, we cannot leave it in water or in air. If we keep it in air, what will happen? It will be dried, the cell will be dying. And if we keep it on water, it will be wet. But what will happen? The water is not isotonic to the cells. This will be hypotonic. The cell will take up the water from and its size will be increased. So it will be damaged, function will be damaged. So what to do? We have to make one saline that is appropriate for amphibian. Our saline is not identical to the amphibian. So for them we make a saline. We call it Ringer solution. Ringer solution. So what is this Ringer solution? How it is made? It is made with NaCl. The percentage in the solution should be 0.6. Very good. And there should be also potassium chloride 0.04%. Okay, so this way you can, and there should be some buffer so that pH is maintained. Sodium bicarbonate and sodium hydrogen phosphate. Okay, so with that we can make a Ringer solution, and some amount of glucose may also be kept in the solution that will supply nutrient to the muscle. But since we will be performing the experiment very fast, so glucose may not be required. Okay, so I have kept the tissue on the Ringer solution. Now I have to make the setup. How I can make the setup? This is the one setup. This is called myograph. This is the stand. You can move up, move down with this stand. And this is a lever. With this lever, there is a lightweight stick. The other end of the stick is attached to, the long stick is attached to, one end is attached to it, and another end is having a writing device. Normally it is a film, X-ray film, cut it and make it arrow shape, then you attach it with the other end of the stick. So now it is the writing device. So when there will be movement of the stick, so this stick will write on the surface of the kymograph. Okay. Now, what has to be done? This is the tendon. So this is the lever, short arm, long arm. So there is a thread, and at the other end there is a hook. I will pierce the hook through the tendon. So this is obliquely now active. This is not resting on the surface. Only this area, joint, knee joint. So only the knee joint will be touching this. This is filled up with solid wax. Okay, so you can pin up the joint with the wax. Okay, so now what is there? The nerve is there. This is the electrode, two electrodes, stimulating electrodes. Now the nerve, you place the nerve over it. Okay? So this is the arrangement of the myograph with the writing lever and the muscles. So this is the arrangement, and this is called kymograph. This is called kymograph. What is there in the kymograph? In the kymograph, it is having a simple motor. And the motor is having different gears. So, इसका मतलब ये है, you can move the drum at different speed. Like car is having different gears, so that the shaft See, the fan is moving, there is a shaft, so it is moving at a particular speed. So now you can increase the movement of the shaft, decrease the movement. You are having a gear here, there is a switch here. So similar kind of adjustment is there, gear. So with that gear, you can make it faster movement or slow. When we will be doing hard experiment, we need slow speed. When we will be doing muscle experiment, we need fast movement. Okay, so that is the purpose. Now, another thing is there. So this is called Du Bois-Reymond induction coil. Du Bois-Reymond induction coil. Its purpose is, it converts DC current. This current is AC current. AC current. The first, with a transformer, we have to convert this AC current into DC current. Now, Du Bois-Reymond induction coil. So you will see the Du Bois-Reymond induction coil. So you see the image of Du Bois-Reymond induction coil. So there is one primary coil, there is one secondary coil. So connection is given to the primary coil. DC current connection is given. With DC current, the DC current, what is this machine we call? Conversion of AC to DC transformer. So DC current will be produced. Now you can adjust the output. What voltage you want? What voltage? Whether 6 volt, 5 volt, 10 volt, 20 volt, 12 volt. It's now output. What would be the output? You can decide. So depending on the output, so I will send this output current, output voltage to the current to the Du Bois-Reymond induction coil, to the primary coil. So now when it is inserted or connected with the primary coil, now the primary coil will produce a magnetic field. What it will produce? A magnetic field. If I place another circuit, another place over the primary circuit, then what will happen in the secondary? That will be the secondary circuit. In the secondary circuit, another type of current will be generated. That is called induced current. That is called induced current. So now the purpose is to produce induced current. You can, suppose this is the primary coil, it is fixed, and the secondary coil, you can move it along the primary coil. If it is away from the primary coil, the amount of induced current will be less. If you come closer to the primary coil, amount of it will be increased. Amount of induced current or induced voltage will be increased. So we need about 4 to 5 volt induced current. We don't want more than this, but that will be not appropriate for them. It may damage the tissue, and it is sufficient to stimulate the muscle, stimulate the nerve. So that is the appropriate. So nowadays, instead of Du Bois-Reymond induction coil, electronic supply is available. That is called student. Maybe you are having the image of that student stimulator. Student stimulator. In some books, it may be there. Student stimulator. So there you don't need any. Are there any image? Yes. So this is the student stimulator. Okay. So through student stimulator, you can decide on what you can decide on. You will be sending stimulus to the nerve once at a time, only one single stimulus. Or you can make repeated stimulus. Then also you can decide, how long, single, but how long? For what duration? So you can adjust. So you can adjust the intensity of the induced current. and the duration of the current and strength or the frequency or the either single or repeated. Repeated stimulus also we need. We will explain. We will go with this for another experiment. So now, instead of Bois Reymond induction coil, we can nowadays can be used. But nowadays these are also not used since there is no amphibian experiment, live experiment. Okay. So we are just telling how actually it has been done. So now this is the recording. With this, you can start it. It will be starting movement. So there is a drum. That is a cylinder, and outside it you can paste a paper, white paper. Now you have to turn the white paper into black. How it can be done? By smoking. You will smoke and blow you the smoke. Thus it will be black. Not that. This smoking means you have to first fix the drum on a rotor. You fix it in a horizontal rotor, you fix it, and this can be rotated. So drum will be moving. And outside there is a white paper, I pasted it. Now, you have to take a kerosene lamp. Kerosene lamp. Now ignite it, then just you start moving and you keep the lamp at the bottom like this. Move it like from this end to that end. And after some time, the surface will be blackened. So this process is called smoking of drum. Now you move the drum from there onto the kymograph. Fix it on the shaft of the kymograph. So there is tightening device. So this way you can now place. So now see, this is the muscle. This is the lever, short arm of the lever. The muscle is attached with a thread. If the muscle contracts, what will happen to it? It will go move upward. This is the muscle. This is the knee. This is the tendon part. This tendon part is attached to the short arm with the thread. That it is hooked with the tendon Achilles. So now, if I put the stimulus, give current, then what will happen? It will be the muscle will be stimulated and it will be shortened. If it is shortened, it will pull the short arm. As a result, long arm will be moved upward. It will be moved upward. So I have given the stimulus for once. So immediately, for a short duration, immediately after contraction, the muscle will be relaxed. So again, the stylus will be coming down to its original position. And by this, as a result of this, you will get, okay? What is PMC? Point of maximum contraction. So this is the point of maximum contraction. Point of, you have drawn a vertical line. So this is the point of maximum contraction. At this point, there is maximum contraction. So at this time, contraction is over. So during this time, contraction is over. So how much time it took for contraction from here to here? So how many frequencies are there? You tell. 0.01 into 3, 0.03. So now you can calculate. Very simple. You will multiply it by 2.5 or 3.5 or 4.5. Simple. You use the fraction. You guess the fraction. Whether it is half of a cycle or quarter of a cycle. So you can calculate it easily. So this way you can calculate what? Relaxation time also. And by a scale, you can measure the height also. Force. In terms of force, I cannot measure. So only thing I can measure the height. If more force is generated, more height will be there. So force here will represent by height in terms of centimeter or millimeter. So you can measure it using a scale. So that is the. Now, can you calculate one more thing? Rate of contraction means speed of contraction. How can you do that? How can you measure it? It's very simple. Rate of contraction. During the contraction period, during this period it contracted. And how much time it took? CP? CP means PC to PMC. This is the time. So if you divide, this is the time and distance is this. Distance is this. So you divide the distance by time. That will give you the speed. Isn't it? Speed of car. How much distance you travel in a particular time. So how much distance? This much distance you covered in this much time. So measure this distance in terms of centimeter and divide it by the time. So time is in second and it is centimeter. Centimeter per second will be the speed. Can you do it? Simple? Okay. Now I am coming into latent period, cause of latent period. Number one, I place the electrode here. Where? I place the electrode here. So in this position, nerve impulse is generated. Then the nerve impulse started conducting to this neuromuscular junction. Definitely, some time will be required to pass the impulse, to travel the impulse from here to here. So, this latent period, after that it will contract. So, this is the traveling time, conduction time in the nerve. So, first is conduction time. Again, the impulse came to the NM junction, but not immediately transferred to the muscle. It will be releasing some acetylcholine. This will be producing action potential on the postsynaptic membrane, means on the sarcolemma. Then it will travel along the sarcolemma. Then it will be entering through the sarcotubular system into the cistern. From there, acetylcholine will be liberated. Then there will be EC coupling. Then there will be contraction. Uri Baba, lot of things. So, what happened? So, one will be the time for conduction. Another is time for crossing neuromuscular junction, and time for EC coupling, and time for mechanism of contraction. So all these plus another thing is there. This end is connected to the liver, short arm of the liver. This liver has got some weight. The liver has got some weight. So it has to lead this. If no weight, it will immediately contract. If more and more weight is there, it will be delayed. So this inertia of the stylus, writing arm, is another reason of delay. So what are the reason of the delay? Number one, conduction. Number two, conduction, to travel from the point of stimulation to the neuromuscular junction. Means from here to here. Then the time is required to pass the neuromuscular junction. Then to make AC coupling, time will be required for AC coupling and time will be required to initiate the contraction. Plus another factor, that is the inertia of the lever, right index. So these are the cause of latent period. This is the delay. This is the cause of the delay. Immediately after application of stimulus, immediately it is not responded. It responded after certain time. Understand this? Is that okay? Right? Okay, fine. Now, suppose this is the twitch you got. Now you have to make a table. You have to write the result. First, what? You have to write A to B. What is this? A, you have to write what is A? Point of stimulation. What is B? Right. What is C? Point of maximum contraction. And C, point of relaxation. Now there are how many periods? 3 periods. One is latent period, one is contraction period, and one is relaxation period. Now in your copy, you write what are the periods. Actually, in second, you write. And you know the each frequency means 0.01 second per cycle. Now you calculate and tell me the results. Okay? Up to this. Here, from this peak. Very easy. You tell your answer. Latent period, he will tell the latent period. Others, please check whether he is correct or wrong. Tell.
Latent period is 0.01.
Is that, say, hey, tell the time. So unit.
Second.
Yes. 0.0. Is that correct? Correct? Yes. Latent period. How much is the latent period? 0.01. And, okay, now in this way you can calculate these periods. Okay. After that, our next experiment is effect of temperature on skeletal muscle contraction, on simple muscle twitch. Okay, effect of temperature means I will apply. First, I will take a normal contraction like this. Then I will apply some cold saline, cold ringer saline, ringer solution. Okay? And after that, after applying it, the muscle and nerve will be cold. It will cool down. Now, what I will do, I will make another contraction. I will give stimulus. Now, contraction will be there. What has happened, you have to note down. What happened to the latent period? What happened to the contraction period? What happened to the relaxation period? What happened to the height of contraction, means force of contraction. Again, I put some normal saline to bring it to normal room temperature. Then I put some hot saline, say 38 degrees centigrade. The earlier is 0.8 degrees, 8 degrees centigrade. Now this is 38 degrees centigrade, more than room temperature. Now then you will get a contraction. Now again you see the observation. What has happened to the latent period? What has happened to the height of contraction? What has happened to the contraction period? What has happened to the relaxation period? Okay?